Hi everyone, and welcome to the Spirometry and Respiratory video lab lecture for you. This video will cover a brief overview of your brain regulatory centers, the receptors that are involved in regulating respiration, and we'll get into spirometry itself, probably most importantly, in regards to your information here, as far as the anatomy, physiology that we'll be assessing and looking at, but as well as different influences that factor the results in your spirometry lab or things to consider. Again, if you're going through your lab manuals, as you should, there's lots of supporting information there. There's some really great videos associated with this lab from outside sources, so definitely review that. And yeah, let's get into it. So to start off with, again, just hopefully this is a bit of review from the other content that you've already viewed. But we can start off with our brain center. So of course we'll have our brain here. It's very anatomically correct, of course. But primarily what we're interested in when we're talking about control centers generally, or something that might be a bit new, is this section in here, right? So the lower brain stem, right? And that's, of course, roughly where the pons is located. Okay. And pons, if you recall, is the bridge, right? So what we'll do is we're going to blast that section out and do a bit more of like a close-up view of that for you, so that we can look at a little bit more of the centers involved. So what we have from here is I'd say three, even though it's kind of maybe illustrated looking like it's four. Okay, so we have this group of nerve centers here, which is called the pontine. Pontine kind of helps you remember where it is. Respiratory group or center, depending on how it's represented. We'll call it group here, PRG. And so you can kind of think of this as one center or two, but they have slightly different roles, the two different parts. So the more superior part here is going to be your pneumotoxic center, and this is your how do I spell that again? Taxic center. Okay. Okay. Sorry. So with that, these influence a couple other centers that you've probably come across already, and that would be the ventral, and let's go dorsal respiratory centers as well. There we go. That's better. Okay. So ventral, you can say respiratory center or respiratory group. And then this would be the dorsal respiratory group. Okay, so now that we have a bit of a lay of the land here, we can start saying how they're influencing on one another, right? So ultimately, the pontine respiratory group, what that's involved in is really modifying the other two centers. So, for example, our pneumotoxic will have a reducing or inhibiting sort of feedback function, whereas the apostic will have a positive influence on that, and our positive drive as far as trying to stimulate respiration. Okay, and again, when we look at the pneumotoxic center, it really regulates the amount of air that we get with each breath, and will limit signals to other nerves like the phrenic nerve to adjust the rate or depth of your breathing. So again, that kind of has that negative inhibitory kind of stimulation or response, whereas the apneistic respiratory group promotes inhalation and constantly stimulates. So we call that kind of like respiratory tonicity. So this constant tone. We have a similar thing from our brainstem in the same region where it keeps tone through our body, right? A lot of the times people think having a toned body in common layperson language means like fit and lean, whereas specifically more to do with your physiology, having tone in your muscles means your brain essentially holds your body up without thinking, right? So it's that low level activation of your musculature. In this case, specifically with your respiration, this comes from the apneistic center of the pontine respiratory group, and it just constantly keeps you breathing. You can think of it as like the pace setter neural mechanisms that we have for our heart as well. So they're very similar in that sense. Okay. Okay. And so again, just kind of continuing on that regulation, what we'll see is, for example, the ventral group goes and innervates our musculature of typically the accessory respiratory muscles, and so like we'll get to those in a moment, and that will help again with the rhythmicity of our breathing, but because it's the accessory muscles that they attach to. So if we have our ribs here, our sternum, and of course there's the intercostals in between. Just as one example of accessory muscle groups are intercostal muscles. What that will do is when we have forced, and this is going to be important as we move forward, forced respiration, that's a main stimulator for that. Okay. On the other side, for the dorsal group, that's our dorsal respiratory group. That's more when we're just thinking about how do I say this, like quiet breathing. So in lab, when we measure something called vital capacity, I'll bring this up again, our quiet breathing is simulated primarily from this. And again, our dorsal group will go primarily, one of its sources is the diaphragm, and different version of the lung, same set of ribcage here, just to show that it's slightly different innervations. This would be more for like the external intercostals, and again, this would be internal primarily, as well as the diaphragm, right there. And again, so that's more for quiet breathing. Inspiration is the primary cue for that, and that's what that innervates with. Okay. Now, as far as all physiology, like when we're talking about how our physiology works, the main overarching theme, the umbrella theme always with physiology is homeostasis, and in particular regulation of. I mean, our body is a set of feedback loops, and without it, things could get out of control pretty quick. But as we know, human body keeps everything tightly regulated. So keeping that in mind, when we have our respiration controlled, I'm not even going to draw for this one. What we have is a handful of receptors, and specifically for feedback, and they include chemoreceptors, baroreceptors, stretch reflectors, and a new-ish one for our lungs that are termed irritating kind of receptors. So usually, chemoreceptors, that's primarily for quiet breathing. So the primary driver of that being carbon dioxide, but of course there's oxygen and pH that is picked up from there. Baroreceptors are also located in our aortic and carotid tissues, and they'll sense changes in pressure relative to the respiratory centers. In addition to that, the more interesting additional ones that I wanted to add into these would be stretch receptors and irritating receptors. And irritating receptors typically are to do with physical or chemical stimuli, but with respiration in particular, and where it comes up is things like asthma, for example. So this is kind of the big point, but also I guess other respiratory things, pathologies. So with the stretch reflectors and the irritating receptors, what will happen in the nasal cavity, the larynx, even down into the branches of the brachial tree, we get this kind of like reflexive protective mechanism that occurs. So we'll cough or sneeze things out that are irritating our lungs and airway passage. And what will happen sometimes with asthma in particular is there's a propensity for some people with asthma that tend to cough more frequently, and both as a result of having an asthma attack, but generally it's found that there's a bit of a genetic link too, to just having a certain receptor be more readily active. So just kind of an interesting side note, fun fact there. Right, so getting back to our rib cage and our lungs here, I'm going to draw a very poor version of our rib cage. Here we got our sternum, okay, and of course in between and above what we have are some respiratory muscles. Now we spoke about what innervates and drives them up here in all of those scribblings and scrawlings, but down here the main one always that we want to think of when we're thinking about respiration is our diaphragm, and of course that sits deep or in the center of our rib cage and attaches near the bottom of them. With that, that's essentially one of the principal drivers for respiration. And so what will happen is when we go to breathe in, what happens is the diaphragm decreases. So let's even just say maybe inspiration here. We'll say, let's use a different color when we get there. Sure. Inspiration. Okay. So with that, again, with inspiration, the diaphragm contracts and lowers, and that's driven from our respiratory centers here, in particular the dorsal respiratory group. So it descends. When that happens, depending on the need and the stimulation, usually our, let's go this direction. I'll say the external intercostal muscles also contract, and what they'll do is they'll lift the rib cage. So our ribs kind of, if the movement of it is the rib cage lifts up this way, providing more space and volume within the rib cage, and the diaphragm of course drops and creates more vertical volume. So this is kind of I guess you could think of it as a little bit more kind of like expanding out, even though the ribs are moving up, and this expanding down to help with that. Okay, and again, that's primarily in quiet breathing when we're just sitting and resting with that volume. Okay, let's keep with quiet breathing. So on expiration, when we're quiet breathing, essentially it's just a recoil of the musculature that drove inspiration that causes the expiration. So again, the diaphragm will essentially return back up. It'll recoil up. The external intercostals will relax, and the rib cage will kind of close down or descend a bit, so that'll give a little bit of an inward closing that way and an upward closing of volume, right? It's the volume of our lungs that way. Okay, great. Let's pick slightly different colors. Okay, so with inspiration, we can also force our inspiration and our expiration, right? Let's go forced. Okay, and that changes things a little bit. One of the main things is the recruitment of more accessory respiratory muscles. Okay, so one of the main ones on inspiration would be more recruitment of what's already occurring, but our accessory muscles include the sternocleidomastoid, and if you get a little bit farther more interior, your scalenes. Okay, those are the main ones, and and that's kind of the main addition. So what they do here, essentially at the top of the ribs, on your first and kind of like sternum, collarbone area, essentially is that it just again helps pull up. So it adds to vertical volume, similar to like the diaphragm did down here, but just from the top end now. Okay. So again, I guess that's the color I was using. So it helps increase some volume that way. Okay. Again, so that's inspiration. When we're forcefully exhaling, let's go to some cool blues on that one. So this is forced exhale. What will happen again is further recruitment of the muscles we use to exhale anyway. So in particular, the internal intercostals now get recruited. And I didn't mention it before, but there's two components of the internal intercostal muscle group, but just for clarity and simplicity, this is probably one of the easier, best ways to do it. So we got internal as well as the abdominals, okay? And that includes the rectus abdominals, the transverse abdominals, and the obliques, which is the internal, external obliques as well. Okay, so we have all of those being recruited to help out, and again, their attachments, of course, are kind of like bottom of the ribs. So when they contract, they essentially help close again. So this is on expiration, it closes the side, the abs here, they pull, and it's a little tricky in 2 dimensions to see. They essentially pull down and in, okay, and kind of like inward. So I guess another way to kind of think of that, I'm going to get rid of those, I don't like that. Another way to say it is they more or less help add to the diaphragm by pushing up and inward at the bottom of the rib cage. Okay, you can kind of picture that and feel that yourself. You really exhale, almost do an ab crunch while you're doing it. You feel that push up and in, and the closing of the bottom of the ribs. Okay, so that's a little bit of the musculature that's involved in quiet and forced inhalation and exhalation. Okay, so main things that you'll want to remember is to do with forced exhale, because it's kind of the new different one on top of passive, and that's one of the spirometry tests. And just again up here as a little reminder, our forced respiration occurs from the ventral respiratory group. Okay, so with spirometry, ultimately what we're trying to measure, well, what we do measure is different changes in, one is a flow rate, so how much volume is exchanged over time, but also total volume as far as our inspiration and expiration. There's three main volumes that we'll be kind of looking at and assessing, but there's lots of other output data that you'll get that you might want to explore in your lab reports, and that definitely do have value depending on what you're trying to assess and look at with someone's pulmonary function. So if we're looking at spirometry, when you get to the lab, or if you haven't seen the videos yet, essentially it's a machine with a mouthpiece, and you blow on it, and there's a fan, and based off of the amount of speed at which the fan blows, that will dictate and measure or infer how much volume of oxygen or air that we're able to inhale or inspire and expire. Okay. And so we've probably, again, looking at your information, you've likely come across something like this already. So this is our volume part of the graph, if you will. That's in milliliters. 6,000, about 3,000, and this will be, I guess, 0. Yeah. Okay, so if you were to have this mouthpiece from the spirometer in your mouth, and you would just sit there as you are now, but you have the mouthpiece in, you probably notice some sort of breathing that kind of looks like this as you undulate between your breath in and breath out. Okay, it's very, very chill. Then if you're to inspire or breathe in as much as you can quickly, you'd see a big spike in the volume at which you've breathed in, big breath out all the way down. Let's say to about there. And then of course, if you return to normal breathing, it looks something like that. Okay. So what are we looking at here? Well, one of the first things that you can recognize probably would be along this level. We could, of course, call that tidal volume. So that, again, that's just naturally as we're breathing, as kind of dictated and controlled based off of the feedback cycles of our dorsal group and the pontine group. Okay, but then we get into something where we have this component where we had that big forceful breath in, then we breathe all the way out, and we had that we had that big breath out, and that got us down to here. Okay, so this would be between our tidal volume and that top kind of peak of what we had. There's a few things, so we could call it inspiratory reserve volume. Now, if you just think about the wording, inspiration or inspiratory, so how much you breathe in, reserve, you can think of it as like the reserve between tidal volume and what potentially is reserved for inhaling. And then that's a volume, of course. And then on the other side, between the tidal volume and there is, of course, the expiratory reserve volume. Okay. And again, as the name suggests, as you breathe out, that's on the other side, right? Okay. So if we're going to look at our vital capacity, so that's one of the first things that we measure in lab, not necessarily the first thing we look at when we're analyzing things, but your vital capacity. So the capacity that we have essentially to do any breathing will be from the maximum amount that we can breathe in, including the tidal volume, to the maximum amount we can breathe out. So that's our vital capacity. Okay, and all of these typically just get into an acronym of just the letters that compose them. Okay, so that's VC, vital capacity. So the one thing the vital capacity does not include is this volume between 0 and that bottom point of our expiratory reserve volume. So this here is essentially just dead airspace that we can't use and can't get to, and say, unless we were like in a vacuum room where it would get sucked out. So typically, to figure it out, we use algorithms and different equations to assess for that. And that is our residual volume. Okay, there's that space in there. Okay, so vital capacity is all the possible air we can breathe in and out, with not including the residual volume, which we can't breathe in or out. It's just there. All right. So the next thing that we measure is forced expiratory volume. So instead of VC, it'll be FVC, so forced. And that typically will be a greater amount than our vital capacity because it's forced. But, it'll be similar in what we see, except it's force and can give us a little bit more information, such as this, where our forced expiratory volume 1. Okay, but before we move on from this graph, the idea is in these two conditions, and why they're going to be similar but not necessarily the same, is vital capacity will be sitting there like a relaxed breath out. So it's a full breath, but not a forced breath, okay? And so for some people, that will be the same or different than your forced expiratory volume. So the idea is, with the forced expiratory volume, it's a forceful, quick breath in. So this part, quick and forceful, followed by, again, a very quick and forceful breath out. And the idea is we're trying to see what your max capacity is as far as the rate at which you can exhale or expire at that point, okay? So, right, how quick and hard can you breathe out? And the 1 here on this term represents in 1 second. We have an FEV3, FEV6, and essentially it's 3, 6, or 1, 3, and 6 seconds. They all tell you different things, but the FEV1 is really the crucial part that we look after, or look for when using spirometry. Now the tip off here, the point that I want to cover before we get going on, is just a little reminder that when we're using forced expiration, tie that into what you know about the anatomy and the regulation, right? So again, if we're talking about forced, we're talking about ventral group, we're talking about your intercostals, your recruitment of abs, sternocleidomastoid massoid on the inhale before we get going. So with all of these recruited, you should, sorry, the dog's barking. You should be able to get more, hypothetically, right? If we're just relaxed versus forcefully trying to get air in or out, you think you would get more. So you will see a little bit of a difference at times between your vital capacity and your forced vital capacity, but ultimately they should be recruiting more or less the same amount of volume. Okay, so just in case you end up seeing some weird data in some of your labs that didn't line up with that. Okay, great. So ultimately, things that will affect this, our volume and our different readings, there's a few of them. And the main ones we account for and take into the machine to figure out the algorithms, because you're going to get a predicted number, volume, percent, or a volume that you should be able to do off of a few factors, okay? And one of them is age. Yeah, we'll start with that one. So age, as we get older, based off of just our lung tissues or and their pliability or different factors with their surfactant levels, etc., as well as the mechanical features of our lung system and degradation of our nervous system, essentially we see a decrease with age as far as, predicted, this would be like our both in our vital capacity, but forced vital capacity. And again, you can think of these capacities as literally is the capacity of the lungs, the total volume of lung that we can inflate and deflate, right? So total amount of lung, and that makes sense. You know, if you start thinking about it, as we get older, that goes down, again, for a number of different reasons. So we'll definitely plunk in that. The main ones that we'll be looking at that age is affected by, is FEV1, okay? So those will definitely change with age. Another influence on some of the spirometry readings we'll see is height. And this comes into play in a few different ways, but one of the main places we'll see is vital capacity is changed, forced vital capacity, and even the FEV1 will change. And if you think about height, for example, the taller someone is, the taller volume they'll have in their lungs, so that's proportionate, right? As well as just general size. So the width of their lungs are going to be wider as well. Okay, so that will change the capacities. But the forced expiratory volume 2 will typically be more challenging because they have more distance to cover in that first second when they go to expire. Okay, and this will actually, taller individuals are going to experience a greater decrease in lung volume as they get older. Okay, so again, just some of that pliability and muscular contraction, because they're so tall, this generally decreases that quicker. Posturally decreases with, say, changes in bone density and posture and compression as we get older. Okay. Another factor that plays in is weight, although this one is more so to do with like functional residual capacity and expiratory reserve volumes. So it's more on the on the exhale side of things that you'll see those go down. Again, we don't specifically look at them, but if you see the values FRC or expiratory reserve volume, so those will tend to decrease as weight goes up. Okay, and the reason for that is just compression and pressure on the lungs keeps the lung volume at a smaller size. Okay. Another that we look at, and this one's a little, I don't want to say contentious, but it's the reason, okay, so there's differences in sex, and one of the main justifications for this difference is for men, they have a longer air passage essentially to get to. So the longer airways typically will lead to decreased FEV1 as we spoke to. So there's also greater resistance because there's a longer air passage to travel. Okay, so they have greater resistance to overcome. And again, people kind of debate and say that's based off of men on average being taller than women, but anyways, they account for this. Similarly, women also have a greater consumption of oxygen relative to men under similar physical intensity. So depending on what you're trying to measure and when, you might see there's an impact for women on that end too, with increased respiration. Some of them might have greater secondary and accessory respiratory muscles come into play, which can affect their inspiratory respiratory volume. Okay, right. And last but not least, even with all of these kind of more demographic or anthropomorphic kind of measures, ethnicity still needs to be considered, as there's differences between ethnic groups, and you'll see that there's a selection on the spirometer itself. Okay. Yeah. All right. Well, again, we've kind of covered influences and considerations when looking at a spirometer. We'll talk more about analysis within, but you have some great videos to help you guide through your analysis of your spirometry data as well. Some general capacities, and then how to tie them in both to our physical anatomy and how the physiology of those groups and feedback systems work to help regulate. Okay. Great. Hope you found that helpful, and yeah, look forward to seeing you in lab.